Good morning, my dear students. I am Dr. Walvekar. I am continuing the class yesterday which I had taken regarding the carbohydrate metabolism. We have seen that one in the carbohydrate metabolism, the type of the food, whatever we take, most of it will be containing major carbohydrate products only, and they undergo the digestion and absorption. And also we have seen that one, the parts that are involved, mouth, stomach, intestine, wherein it will be acted upon by the different enzymes. And the idea of that one is to convert that the macromolecular size foodstuffs into the simpler one, and then it will be absorbed in the blood, and then it will be passed on to the bottom of the liver. So once it is absorbed, it will be the one which is considered as a glucose molecule only, because it is a fuel which is acting as the one providing the energy to the body, and further it should undergo metabolism to gain the energy for the body for the various other purposes. So we define metabolism as the one process wherein the body converts the foodstuffs into the simpler one and forming the energy in the form of the ATP, and it is consisting of the two types or parts, phases we can call, which are referred as the catabolism and anabolism. So catabolism is the one which is applied for the breakdown of the complex molecules into the simpler one, and along with that one there will be the release of heat or the energy, or you can call that in the biochemical language as the formation of the ATP. Whereas in the anabolism, it is the formation of complex molecules into the simpler one, which requires the heat. It is something like that applying for any purchasing of the shares, they have to first invest and then take back whatever the profit we get. So first, in the starting of this metabolism, there is a need of the investment or utilization of the fuel or in the form of the ATP, and then there will be more amount of the formation of the ATP. So this is the metabolism, and this metabolism of the carbohydrates has got different pathways. The pathways are about 8 to 10 major pathways are there. Out of that, the glycolysis is the important major pathway which we had started yesterday. We see that it is the pathway which has got the 10-step chemical reactions that take place. Once the glucose is formed, that is taken up in the liver, and it will be converted into glucose 6-phosphate. So till that step we had discussed, and today we will continue with that next topic. So metabolism of carbohydrates largely, you know, well, it is the metabolism of glucose only, because whatever the monosaccharides that will be formed after the breakdown of that one, it will be the glucose only, and the glycolysis is the major pathway of this glucose oxidation. It has got the 10 tests of reaction. So we see that yesterday the definition for the glycolysis, it is defined as a sequence of the reactions which will convert the glucose or the stored from the form of the glucose, that is glycogen, finally forming a product which is in the two molecules of pyruvate, and it can be taking place in the presence of oxygen, which we refer as the aerobic reaction, and also there can be the formation of pyruvate to lactate, which will be taking place in the anaerobic conditions with the simultaneous production of ATP. This one is referred as the embedded layer of pathway. This is the definition what we had given for the glycolysis. So this glycolysis has got the 10 steps. So that so you need, so in some textbooks they are divided into the two phases, but for broader understanding given, it is divided into the three phases or the three stages. One is the energy investment phase or priming stage. Second one is the splitting phase, and third one is the energy generation phase. So in the first energy investment phase, in this one, there will be the conversion of glucose into the glucose 6 phosphate. So that takes place by the phosphorylation. So the D-glucose is converted to glucose 6 phosphate, and the enzyme that is involved in this is the hexokinase or also the glucokinase. We'll tell you about these two later on. So it undergoes the phosphorylation and it yields glucose 6 phosphate. Glucose is converted into the glucose 6 phosphate with the ATP, but ATP is required as the phosphate donor. Then it also one which requires the magnesium as the cofactor for this reaction, and this is accompanied with the considerable amount of loss of the free energy. And another thing is this reaction is irreversible one, because the glucose 6 phosphate what has formed, it is in the cell membrane is impermeable for this glucose 6 phosphate. And what happens in the case when there is the less amount of glucose is there, then it will be utilized by the hexokinase. So glucokinase, it acts only when the levels of glucose is more than 100 milligram per deciliter, and there will be the one consumption of one ATP molecule in this one, and the formation of glucose 6 phosphate in the central molecule that forms, or which is a central place for further the pathways like glycogenesis, gluconeogenesis, and pentose phosphate pathway. So once the glucose is entered, it undergoes the phosphorylation and gets converted into the glucose 6 phosphate in the presence of hexokinase or the glucokinase, and it requires the magnesium as the cofactor, and there will be the loss of one ATP molecule. So here the reaction shows you that glucose with the utilization of the ATP in the presence of magnesium and hexokinase as the enzyme, it gets converted to the glucose 6-phosphate. One phosphate molecule will get attached at the 6th position, and this is the one glucose 6-phosphate is formed, which is this reaction is a irreversible one because the cell membrane is impermeable for the glucose 6-phosphate. Second step, that is conversion of glucose 6-phosphate to fructose 6-phosphate. So this is the second reaction wherein the fructose 6-phosphate is getting converted by the glucose phosphate isomerase. The enzyme that is used here are applied to the phosphoglucose isomerase. The reaction is, as the name suggests, the reaction is taking place as a isomerization of the reaction. That is the same type of the structure, but only structurally it is different one. And please note that this is a reversible reaction. So in the second step, it is the enzyme which is acting upon is the phosphoglucose isomerase. So it is phosphoglucose isomerase. In some text, it may be given as a phosphohexose isomerase also. So glucose 6-phosphate it is getting converted into the fructose 6-phosphate. Then now the another enzyme that is used in the third step is the fructose. That is the third step. Phosphofructokinase is the one enzyme which is acting upon in this step using the magnesium as the cofactor, and it forms into the fructose 1,6-biphosphate. That is from the fructose 6-phosphate to fructose 1,6-phosphate. Similar to the step that is in the glycolysis first step, the second molecule of the ATP will be utilized, or the ATP will be provided here, and this step is irreversible reaction. See, the glucose 6-phosphate by the action of this PFK in presence of magnesium and provision of the one ATP, it is getting converted to fructose 1,6-bisphosphate. This is the third reaction that is occurring in the glycolysis. Coming to the second stage, that is called as the splitting stage. There will be the formation of two molecules that are the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This will be formed by the action of adlose. This one, it acts upon this fructose 1,6-phosphate, and these two components will be formed. So here this one is referred as the splitting phase. So fructose 1,6-bisphosphate by the action of aldolase, it forms to the two components, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And then one enzyme in there, step number 5, the enzyme trirose phosphate isomerase, it converts dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, so that every molecule of the glucose entering the glycose, 2 moles of or the 2 molecules of glyceraldehyde 3-phosphate will be formed. The acting enzyme is trirose phosphate isomerase, which will be acting upon the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So here, one thing to be noted here, that the glyceraldehyde phosphate is the only one molecule that continues in the glycolytic pathway. As a result, all the dihydroxyacetone phosphate produced are acted upon by the enzyme trirose phosphate isomerase, which reorganizes the DHAP into the EAP so that it will continue in the glycolysis. So remember that only the GAP is getting continued in the glycolytic pathway, but DHAP is get mixed or reorganized into the GAP, and it also will be continuing in the glycolysis. So in some text, or can call that as a preparative phase. This finishes with the first part or the major part of that one, preparative part, or you can call the investing part, like that one. So once those things are formed by the enzyme called trirose phosphate isomerase, this will be acted upon these two, and dihydroxyacetone and glycerol 3 phosphate are formed here. And in the sixth one, as I said earlier, that glycerol 3 phosphate is the one which is continuous in the glycolytic activity only. So the enzyme glycerol 3 phosphate dehydrogenase, it catalyzes and adds an inorganic phosphate to glycerol 3 phosphate, and it forms into the 1,3-bisphosphoglycerate. You remember that in all these reactions, they have done one good thing by naming the similar name of the enzyme, which will be something very closely related to the product that will be formed, so that we can remember it easily. So the enzyme glyceraldehyde 3 phosphate dehydrogenase is the one enzyme. It catalyzes the glyceraldehyde 3 phosphate and converts it to the 1,3-bisphosphoglycerate, and there will be building of NADH along with that one with a hydrogen atom. So this step is very important because there will be the formation of NADH, and in aerobic condition NADH passes through the electronic chain reaction, and 6 ATPs are synthesized by the oxidative phosphorylation. So it is a reversible reaction. You have seen that one in first and third steps. There was the investment of 2 molecules of ATP, and here there are formation of 6 ATP molecules. So glycerol 3 phosphate, in the presence of this GAPDH, it gets converted into 1,3-bisphosphoglycerate, and there will be along with that one, there will be the formation of NADH also with the hydrogen atom. Seventh one is the position. The step 7th is phosphoglycerate kinase. This is the name of the enzyme, and this step involves the loss of the phosphate group from the starting material. This phosphate is transferred to molecule of ADP that gives the first molecule of the ATP. Since there are 2 molecules of 1,3-bisphosphoglycerate, this step actually synthesizes the 2 molecules of the ATP. The importance of this step is the formation of the ATP, wherein there will be the enzyme which is called as the phosphoglycerate kinase, which is acting upon this 1,3-bisphosphoglycerate. So this step is also called as the substrate level phosphorylation, as ATP will be forming here, and with this formation of ATP, so you can nullify what the first two ATPs molecules are formed in the first positions, or first and third positions. So it amounts to the utilization and gaining of ATP molecules amounts to 0. And here again, the magnesium is the one which is involved in this reaction, which is acting as a cofactor. So it is the 1,3-bis phosphoglycerate is converted into the 3-phosphoglycerate, and there will be the formation of 1 ATP. From the ATP, so the one phosphate molecule will be added. As because there is the two molecules, the two molecules of this phosphate will be taken into the account, and two ATP molecules will be formed here. So now coming to the 8th stage, which is consisting of the enzyme action by the phosphoglycerate mutase. This enzyme catalyzes and relocates the phosphate from 3-phosphoglycerate from the third position to second carbon of the 2-phosphoglycerate. The action of this one is to relocate the phosphate from 3-phosphoglycerate to the second form, that is called as the 2-phosphoglycerate. It is just a simple rearrangement of the position of the phosphate group and making finally as the 2-phosphoglycerate. So mutase is an enzyme that catalyzes the transfer of the functional group from one position of a molecule to another. So the role of phosphoglycerate is to relocate or rearrange the position of the phosphate from the third phosphoglycerate to the second carbon of the 2-phosphoglycerate. And this mechanism, it continues by addition of the additional phosphate group at the second position of the third phosphoglycerate. You can see this one only when you see the diagram. From the third position of the phosphate one, which is reshifted to the second position, and this 3-phosphoglycerate, it is getting converted into the 2-phosphoglycerate in the presence of phosphoglycerate mutase. The function of this mutase is to relocate or rearrange the phosphate group. So from the third position, the phosphate is relocated to the second position. That is in the step number 8. And now in the step number 9, the one enzyme that is utilized is enolase. This step involves the conversion of 2-phosphoglycerate to the phosphophenol pyravate. This is the place where the 2-phosphoglycerate is converted to the phosphophenol pyravate. This removes or dehydrates the molecule of water, and it forms phosphophenol pyravate. One thing I have to add here is that in the laboratory, for the collection of the blood samples for the sake of processing the blood glucose level, blood will be collected with the anticoagulant. So the anticoagulant is this potassium oxalate, and also it will be added with the sodium chloride. So the idea behind using the sodium chloride is that it inhibits the action of enolase. It inhibits the action of enolase. That's how what happens. The glycolytic activity will be stopped. Means whatever the amount of glucose will be present in the got sample will not undergo glycolysis process, and the glucose whatever it is present in the blood sample, it remains as it is. So whenever we collect the sample in the test tube containing the potassium oxalate as an anticoagulant, which will not allow the blood to clot, along with that one we are also adding sodium chloride. The sodium chloride is the one which inhibits the action of enolase enzyme. So that what happens? The conversion of glucose into the further activity will be stopped. So the loss of glucose will not be there in that sample. So the 2-phosphoglycerate by the action of enolase, it forms into the phosphophenol pyruvate with the formation of one water molecule. Now coming to the last step, that is pyruvate kinase. It is a step which is a final one wherein the phosphophenol pyruvate, it gets converted into the pyruvate with the help of the enzyme pyruvate kinase. This is the end product, that the pyruvate that forms from the starting molecule, that is glucose, from glucose to pyruvate. This enzyme pyruvate kinase, it transfers a good phosphorus from the phosphophenol pyruvate to the ADP and forms the pyruvate or pyruvic acid, and along with that one the ATP will be formed. So there are two molecules of the PEP, the reaction generates the two ATP molecules. So this step is called as the substrate level phosphorylation, and again it requires the cofactor like magnesium or manganese along with the potassium, and this step is the irreversible reaction. So the last step, that is the final step, wherein there will be the use of pyruvate kinase. So that the pyruvate kinase converts phosphophenol pyruvate to the pyruvate. So all this, it happens in the presence of oxygen, means formation of oxygen, the conversion of the glucose into the pyruvate. Till that we call that as the glycolytic pathway, which has taken place in the aerobic condition. So phosphophenol pyruvate getting converted into the pyruvate in the presence of pyruvate kinase, and also there will be the formation of ATP. So in short, you can see that, visualize this one. Glucose is converted into the glucose 6-phosphate, and then you can remember this thing, as it may be a very hard one to remember it. This is a simple step I am showing you. You can remember the final products that are formed: glucose 2, glucose 6-phosphate, then fructose 6-phosphate, and then fructose 1,6-phosphate. Then there will be the splitting of the molecules will be taking place, and it forms into the two things, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Two types of these molecules will be formed at the 4th position, and then from glyceraldehyde 3-phosphate, it is the one which continues in the glycolytic pathway. The dihydroxyacetone phosphate, it gets rearranged and acted to continue with the glyceraldehyde 3-phosphate only, and glyceraldehyde 3-phosphate, it gets converted into 1,3-bisphosphate glycerine. From there to 3-phosphoglycerate, and then to 3-phosphoglycerate. Two molecules will form. From there, it will be phosphophenol pyruvate. From 3-phosphoglycerate to 2-phosphoglycerate, and then to phosphophenol pyruvate, and then to the pyruvate. This is the last one. That after that, the end product formed is pyruvate. So these are the steps. This way you can remember at least the products that are formed. You can see once again: glucose to glucose 6-phosphate, then glucose 6-phosphate, glucose 1,6-phosphate, and then two molecules like this: dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then 1,3-bisphosphoglycerate, 3-phosphoglycerate, then to 2-phosphoglycerate and phosphophenol pyruvate, and then to pyruvate. These are the 10 steps wherein there will be the pathway of the glycolysis involving the 10 steps. So the enzymes that are used at all the 10 steps. First is hexokinase or glucokinase. Second one is phosphoglucose isomerase, and third one is phosphofructokinase. Fourth one is the aldolase, and fifth one is trirose phosphate isomerase. Then sixth one is the glyceraldehyde 3-phosphate dehydrogenase. Seventh one is phosphoglycerate kinase. And eighth one is phosphoglycerol mutase. And then a ninth one is the enolase. And I told you regarding the inhibition of this one can be done by the addition of the sodium chloride and then pyruvate kinase. So these are the 10 enzymes they get involved in the glycolysis glycolytic pathway, and the products that are formed are at the end it is 2 ATP, 2 NADH and 2 pyruvates. So how these 2 ATPs are formed, we'll show this in the next slides. Still, in the one slide only it has been given here how this glycolytic pathway is there. The hexokinase, 6-phosphate, and then fructose 6-phosphate, fructose 1,6-phosphate, and finally pyruvate. So you can write it several times or the way you want, then only you can remember these reactions. The flow chart is sometimes they may ask in the question in the examination, write the 10 steps of the glycolysis. So that time you can remember and write it this way, at least the namings of this one. So glucose to pyruvate. Now in the first step and the third step there will be the utilization of 2 ATPs will be there, and in the 7th and the 10th steps there will be addition of 4 ATPs will be there. So finally the amount of ATP that is made available is the 2 ATPs in the end of the glycolysis. So here to be more elaborately informed here, ATP formed are this way in the 3 reactions, and ATP whatever it is consumed, it is one from glucose to glucose 6-phosphate, there will be consumption of 1 ATP, and then fructose 6-phosphate to fructose 1,6-biphosphate. So that time 1 ATP will be lost. So finally what happens? The 2 molecules will be there. However, the synthesis is 8 ATPs will be there. So what happens when once the glycolysis is finished, the cell must continue the respiration in either aerobic or anaerobic condition. Aerobic means in the presence of oxygen, and anaerobic means in the absence of the oxygen. Under aerobic conditions, pyruvate is transported into the mitochondria and undergoes oxidative decarboxylation, and it forms acetyl coenzyme A and further oxidation to carbon dioxide in the citric acid cycle. The citric acid cycle is the central pathway which is getting involved with both the glycolysis Krebs cycle, or it may be coming from the gluconeogenesis, glycogenesis. All these things will be getting surrounded over or getting mixed with the citric acid cycle, where there will be the formation of acetyl coenzyme A, and this will be getting converted to formation of carbon dioxide and water molecule along with the existence of the ATP molecules. So now coming to aerobic glycolysis. So the tissues that function under the low oxygen condition, they produce the lactate. So in the absence of the oxygen or very less amount of the oxygen is available, if the continuation of the glycolysis takes place, they form the lactate. Means up to formation of pyruvate, it will be considered as the aerobic one, and if there is the unavailability of the oxygen, then that will form the lactate one. So these things takes place in the muscles like contractile skeletal muscle or certain cell types that in retina, they convert the glucose into the lactate even in the aerobic condition because there won't be presence of mitochondria. The lactate production is increased in the septic shock where there will be enough amount of the blood will not be there or supplied there, and these will be the one reason where there will be the formation of lactate takes place. And this formation of lactate can also be found in the some types of the many cancers also, where there will be the more demand for their growth and they require more energy from the glycolysis. So that time when there is more amount of the glucose is required, then this condition, the glycolysis that finishes is called as the anaerobic glycolysis because the product that will be formed will be the lactate one. So now that's what we see, that one, that is conversion of the pyruvate to lactate. So it depends upon the presence or absence of the oxygen in the cell. So in the anaerobic condition, pyruvate is reduced to NADH to lactate by the NADH to the lactate. And the NADH utilized in this step is obtained from the reaction catalyzed by glycerol 3-phosphate dehydrogenase. And this formation of the lactate allows the regeneration of the NAD, which can be reused by the glycerol 3-phosphate so that glycolysis can proceed even in the absence of the oxygen to supply the ATP. Means what happens in this, when there is no availability of the oxygen, the NADs is the one which is converting that lactate to the glycerol 3-phosphate so that there will be the continuation of glycolysis and also there will be the production of ATP. So you can see here how this lactate dehydrogenase is the enzyme which converts this pyruvate into the lactate one. So the question may arise that when there is no provision or availability of the oxygen, then what is the need of the glycolysis to be continued? But the uninterrupted glycolysis continuation, which there is much essential for the some tissues that they function under the hypoxic condition which produce the lactate. For example, the skeletal muscle, smooth muscle, when there will be under the heavy exercises and the erythrocytes, they will be lacking the mitochondria, then they are bound to follow this anaerobic glycolysis. So the formation of lactate will take place in the tissues or the cells of these skeletal muscle, smooth muscle and the erythrocytes. However, after some time, elevated lactic acids levels. So when there is the more amount of the lactate is formed, then they have some the abnormal effect will be there that they lowers the pH value of the blood. The pH value of the blood is 7.35 to 7.45. So when the patient is having this acidity or the formation of lactate will be there, lactic acidosis we call that one. So there will be the another finding will be that the decrease in the pH of the blood will be taking place. So as such, lactate has no metabolic role, and it is in fact a toxic one. And if it is happening formation of the more amount of the lactate, it leads to the muscle fatigue, cramps, or even the lactic acidosis. These are the clinical conditions which can happen if there is more amount of the formation of the lactate will be taking place. So the rise in the level of the lactic acid in the blood, the normal range is 4 to 15, will be there, and mild forms of the lactic acidosis are associated with the strenuous exercise, shock, respiratory diseases, some COPD cases. COPD is chronic obstructive pulmonary diseases. So it may take place, and if there is a very severe forms of the lactic acidosis that can be found in the myocardial infarction cases or pulmonary embolism or the shock condition or uncontrolled hemorrhage. In all these conditions, we may find the glycolysis is taking place in anaerobic conditions, and the end product that is formed is the lactate one. And because of this one, there will be the drastic reduction in the ATP synthesis will be there. Now, coming to this lactic dehydrogenase, that what lactate formation will be there, that is considered as the LDH, one enzyme, that will be one take place or elevate in the conditions where the patients are suffering from the MI. So in this one, what I want to emphasize on one fact that this the LDH is the one enzyme, it gets elevated or it starts elevating after the 24 hours, and the peak level will be in the 3 to 6 days, and it will be there up to elevation level up to the 13 or 14 days. The importance of this one is that one, if a patient or a person is happening to be away from the hospital or in the peripheral area, the villages, and it is not able to attend or to get the hospital facility, and by the time he comes to the hospital and the doctors who will be treating him, if they check for the LDH activity, he can confirm whether that person had the heart attack or not, because some enzymes which help in the confirmation of the MI with the patients is the enzyme called CPK or CKMB. We call it CKMB, that is the isoenzyme, which will be elevating within the 4 to 6 hours, and by the end of the 2 days, it will come down. And if the same patient is happening to come or reporting late to the hospital, that time the estimation of this LDH will help clinicians to make the diagnosis of the myocardial infarction. And then there are two hormones which will be in the role for the glycolytic pathways to continue. The one is the insulin, another one is the glucagon. This insulin, it stimulates hexokinase and glucokinase, converting the glucose to glucose 6-phosphate. And glucagon is the one which is in the liver, which stimulates glucose 6-phosphate by converting the glucose 6-phosphate to glucose and fructose 1,6-phosphate. They will be acting exactly opposite to each other. Now, you must have seen that one. We have used the word like hexokinase and glucokinase, both enzymes in the first step, wherein glucose is getting converted into the glucose 6 phosphate by the action of either glucokinase or hexokinase. So what is the difference between the utilization of these two? We will see here. Glucose first converts glucose 6 phosphate by the hexokinase or the glucokinase using the ATP molecules. But glucokinase is present in the hepatocytes of the liver and beta cells of the pancreas which leads the response to changes in the glucose levels. But the fact is that comparing to the hexokinase, glucokinase has got the lower binding affinity. It is induced by the insulin, and defects in the glucokinase can cause the maturity onset diabetes of the end. That is, we call as the MODI, not this Narendra Modi. This is called as, earlier they used to call that, if the young people used to suffer from the diabetes, they used to call this by the name of this one. What happens whenever there is high level of the glucose is there, there will be definitely the entrance of insulin will be there. That time it is acting upon. So hexokinase is the one which is present in the almost all the tissues, whereas the glucokinase is the one which is pertaining to the liver only. So glucokinase phosphorylates glucose when the glucose is abundant. This activity of the glucokinase is influenced by the carbohydrate intake. It decreases in the starvation and diabetes. What happens when there is no flow of supply of the glucose into the cell is less, then that time the activity of the glucokinase will not take place. Whereas when there is a normal person is there, when glucose is more amount of the glucose is in the cell supplied, that time the hexokinase is the one which comes to the help of the converting the glucose or helping in the glycolytic pathway. When glucose concentration is low, so glucose kinase is inhibited by the one enzyme, protein that is called glucokinase regulatory protein. This protein will not allow the glucokinase to act upon on the glucose molecule, as there will be the concentration of the glucose will be lower. So the liver glucokinase is an inducing enzyme that increases its synthesis in response to the insulin and decreases in response to the glucagon. This is the difference between glucokinase and hexokinase. So I told you that the sodium chloride is the one which gets used in the clinical biochemistry lab, that is at the time of the collection of the blood samples for the estimation of the blood glucose. So this sodium chloride, it inhibits the glycolytic pathway, so because it inhibits the action of the enolase. And then coming to the cancer and the glycolysis. Cancer cells shows increased uptake of the glucose and the glycolysis. This is for their survivability. They need more amount of the ATP molecules or the energy from the body, so that they fasten the glycolysis activity. So that this is the relation between cancer and glycolysis. As the tumor grows rapidly, blood vessels are unable to supply enough amount of the oxygen, and hypoxia condition exists. Due to this, anaerobic glycolysis will be taking place, and as however the cancer cells cannot grow and survive without the proper vascularization. So what happens? That's why when treating the cancer patients, they prepare such a drug which can inhibit the growth of these cancer cells, or demanding for the more amount of the glucose. This is how the cancer patients can be treated by treating them by the drug which will inhibit the demand for the more amount of the glucose or the supplement of the oxygen for that same purpose. This is regarding about the cancer and glycolysis. So in this today's class, we have seen that one. There are the 10 steps are there for the glycolytic pathway to take place. So you have seen that one, that the schematic representation is there, that so from glucose to the formation of pyruvate, you can remember this schematic diagram, and you should remember two things, at least two things, that is the enzyme, which enzyme is getting applied or acting upon at which step, first one, and second one, what will be the product formation that is formed at which step, glucose to glucose 6 phosphate, then to glucose 6 phosphate, glucose 1,6 phosphate, and then glyceride phosphate and glyceride dehydrogenase and 1,3 bisphosphate glyceride, 3-phosphoglycerate, 2-phosphoglycerate, phosphophenol pyruvate, and finally pyruvate. And if there is no availability of the oxygen, it will be getting converted to the lactate. This is how you should remember it, or you can take down the things in the writing one and repeat it. Then only you can remember, because biochemistry subject becomes sometimes the very volatile one. By writing these reactions on and every time, then only you can remember it. Okay? Thank you.
Thank you, sir.
Thank you, sir.
Thank you, madam. Thank you.